So, welcome to my talk. I'm Dennis Röder. I'm curator for herpetology at the Leibniz Institute for the Analysis of Biodiversity Change, located in Bonn, Germany. And during the next 50 minutes, I will present a recent study on butterfly altitudinal range shifts during the last 6 decades. So, as we all know, climate change is ongoing, and if we compare historical climatic fluctuations with the more recent one, we easily can see that we have very accelerated rates of temperature increase, and this has already led to severe impacts on many species and ecosystems. And what we also know is that we are in the midst of recent insect declines, which were in the media during the last years very pronounced. And we lost about 60% of our biomasses in Germany, for example, which leads to many ecological problems. And one of the main groups which is affected are the butterflies. And they are affected because many of them are highly specialized. They have a biphasic life cycle, including a larval stage and an adult stage. Many depend on specific food plants, which makes them very vulnerable. And this is why we choose butterflies as our study species. Our main questions were, have mountain butterflies shifted to higher elevations during the past decades due to climatic changes? And if so, what are the characteristics of the species which have changed? Does it depend on the dispersal behavior, the ecological specialization, or which other ecological properties qualify them as more vulnerable? From the method point of view, we used big database on species records. These were museum records and observations from monitoring programs. And as second line of evidence, we used climatic data, some physiological properties of the mountain environment, such as exposition, aspect, slope, insolation, and developed species distribution models and niche estimates based on hypervolumes for each of the species. From the habitat point of view, most of the species depend on specific grasslands, alpine meadows, calcareous grasslands, but also other habitat types. Our study was recently published in scientific reports, so if you want to go into more details, feel free to read the full article. I can only give a brief overview here. For the climatic data, we used high-resolution surfaces of interpolated weather station data from these two publications of Hebel and Fry. They represent climate data with a resolution of 1 square kilometer and a temporal resolution of 1 month, ranging from 1961 to 2019. So for each of these years, we actually had precipitation, minimum, maximum, and average temperature on this resolution. And I used elevation data to further interpolate this data. The record dataset comprised 11,400 unique records of about 40 species. They had a spatial resolution of 10 meters, so they are very accurate because they were all from monitoring data and from specific collection sites with accompanied GPS data. And the temporal range was 1961 to 2019. The monthly climate data I've interpolated to a resolution of 50 meters because 10 meters was not possible due to the increase in computation times. We had also land cover information with a resolution of 100 meters. This was the ecosystem type map shown below. These are already some of the results. I will play a small video sequence to illustrate the temporal changes. So what you can see here is the elevational gradient in the study area, ranging from about 500 to 3,500 meters. Colored is the potential distribution of the species. These are the models for each of the analyzed years. Below you see the differences of the respective year relative to the average across all years. But you already can see that there are quite strong fluctuations. Maybe let's say give you some more details on the modeling approach I used. So for each of the records for the respective year, I've extracted all the climatic data from the year in terms of bioclimatic variables. This data set was then used to train a principal component analysis. So the principal component analysis comprised all of the years. So it was trained with a full climatic data set and afterwards projected on the original annual variables. So we had for each year, we had the principal components mapped, and these data sets were then used to train maxima models where we restricted the feature ranges to quadratic, linear, and product features, which we think revealed the best fitting response curves. Then we extracted for each of the two data sets, the species records and the distribution models, the elevation data. So we had for the species records, for each record, we know at which elevation the species was recorded. For the species distribution model, I used weighted averages across the probabilities across the species potential distributions. And in the first question, I mapped this, the maximum occurrence in which decade a species was recorded and the minimum altitudinal occurrence in which the species was recorded against each other. And if we would assume that species elevated their ranges to higher elevations, we would see that most of them are in the upper left corner. And this was actually the case in both data sets. We have already some indication that we see at least a trend uphill for most species. The second question was now in which decade were the minimum and maximum recorded. So you see here the observed and the modeled numbers of species with their minimum. These are the light gray bars, and the maximum dark gray bars observation and then per decade. And if you run a simple linear regression across this, we already see quite some strong associations. So we see also using this approach an upward shift. Then we also checked this on for the species records themselves, not looking at decades, but looking at actual years. The same for the species distribution models. For the records, we see quite some noise. This might be due to the fact that the detection of a species, especially on the range limits, might be complicated because they tend to be rare in these higher elevations. So having them in one year or not in the other year could be simply an artifact by sampling. This is somehow counterbalanced if we use the weighted elevational range from the species distribution models. So this could explain why we find a much stronger signal using the species distribution models compared to using the species records themselves. We also looked at the regression slope between the altitude and the decade when the species was detected, and the niche volume of the species as estimated by hypervolumes. Here we also find some significant results. So we see that the climatic niche, the larger the niche is, the stronger is the regression coefficient. So we see that species having a broader climatic tolerance tend to have a higher range shift across the years. Looking at the variance mean ratios compared to the median altitudinal position of the species, we also find some significant correlations. So we see the higher variance mean ratio, the lower is the median altitude, the lower the variance mean ratio, the higher is the median altitude, which gives us also some indication of the niche breadth and the altitudinal range breadth and the influence on the altitudinal changes. Overall, to summarize our results, this is now the video animation of the richness changes. So the warmer the color, the more species are present. Again, we see across all species quite some strong fluctuations. We see a strong range in the elevational changes and also in the richness changes. On the upper right, we see the probability distributions of all species across the elevational gradient. You see that we have most species around 2000 meters where they peak actually. And we see also that the interannual variability is quite strong. So, and with this illustration, I would like to finish my talk, and I'm open for questions.